All right, welcome everybody. Thank you all for being here for the Arkansas Native Plant Society's monthly webinar series. We're very excited to have Caity Sims as our guest this month as we continue to learn about the flora of the islands of the Mississippi River. This webinar is being recorded, and afterwards the recording will be uploaded to our YouTube channel. If you'd like to learn more about the Arkansas Native Plant Society, you can do so by visiting our website at anps.org. Joining is it's pretty simple. You just go to anps.org/join, and you can use a PayPal account to join online, or you can send a check to our treasurer, and our treasurer's address is located there on the website. I'll also be placing links to these resources in the chat so you can find them there as well. For more details about our other webinars and hikes, I encourage you to check out our Facebook page at facebook.com/Arkansas Native Plant Society. We do have the next two webinars coming up in our webinar series. Would be Saturday, September 3rd at 10 a.m. Nate Weston of the Beaver Watershed Alliance is going to be talking to us about biodiversity and the role of disturbance in managing natural ecosystems. And then Saturday, October 22nd at 6 p.m., we're going to get real philosophical. Justin Thomas is going to do a program for us called Thesis, the Nature of Nature. Today, our guest speaker, as I mentioned, is Caity Sims. Caity graduated from Southeast Missouri State University in 2019 with a Bachelor of Science in Wildlife Biology and Conservation. After graduation, she worked at the Missouri Botanical Gardens Litzsinger Road Ecology Center as a restoration intern, where her passion and appreciation for native plants grew. She started her master's in biology at Arkansas State University in Jonesboro in the spring of 2020, and spent the past 2 summers working on lower Mississippi River islands, conducting floras, assessing plant diversity, and tracking the phenological shifts. So Caity will be graduating with her master's degree this December. And so if anybody out there is looking to hire a bright young botanist to help them on with their research or whatever, I'm sure that Caity will be more than willing to entertain some job offers. So with that, I'm going to hand this over to Caity and let her tell us about her research.
All right. Well, thank you for that introduction, and hopefully you all can hear me okay and there's no disruptions. I am at a hotel, like a conference center, so there might be some people walking around behind me, but so sorry about that if that's a distraction. But yeah, so today I'm going to talk about my thesis research, and then just kind of my experience out on the river as well. Okay, so yeah, this picture here on this title slide is really cool. This is of Buck Island, which I'm going to talk about a little bit more, but this is where my research was conducted, and Travis Marsico took this picture on his flight to Memphis a few months ago. So I know you just got a little short description of my background, but I'm going to go into that a little bit more. So I'm from Monroe, Missouri. It's a small city about 45 minutes north of St. Louis. And so you can see that here on this map. So here's kind of the St. Louis area. And then this is where I'm from, Monroe. And you might see from that I've lived close to the Mississippi River my entire life. I grew up living in a floodplain next to the Mississippi River, and have dealt with the effects of its seasonal flooding my whole life. My parents' basement would flood when the river level would get to a certain height, and then I would also deal with some road closures that would prevent me from leaving my parents' house and getting out to the rest of the world. So I'm pretty familiar with the habits of the Mississippi River and what it can do to us humans. So yeah, I graduated from Southeast Missouri State in 2019. And while I was there, I was previously interested in canids and big cats. I thought I wanted to work with large carnivores. And I volunteered at the Crown Ridge Tiger Sanctuary in St. Genevieve for some of my internship hours as an undergrad, and kind of just help cleaning up the facility and making enrichment for the big cats there. And then I did a summer internship at the Endangered Wolf Center in St. Louis. And there was, you can see here is a picture of me doing a paw painting with one of their maned wolves. And so I was just kind of doing keeper duties, cleaning up the facility and preparing diets. And after doing those two things, those two internships, I realized that I really didn't like the zookeeper life, and I wasn't actually that interested in working with animals. So then I took a spring flora botany course as an undergrad with my advisor, Alan Bornstein. And so that's kind of where my passion, and I fell in love with plants, and I got to see plants in a totally different way. And that's kind of where it all switched. So I went from wanting to work with big, large carnivores to studying plants pretty quickly. And although at the time I developed, or I didn't know it at the time, but I developed an interest in phenology through that spring flora course. But I didn't know what phenology was, or that you could even study it at the time, just because from that year I was excited to look for spring ephemerals, and I'd always be checking iNaturalist to look for them. And so I was already studying the phenology of my local spring flora, and I didn't even know it. So then after I graduated from Southeast Missouri State, I did an internship with the Litzsinger Road Ecology Center with the Missouri Botanical Garden, and I was a restoration intern. And then part of my kind of duties and responsibilities of that internship was to conduct a summer research project. And so I decided to study the algal diversity in the creek. It's kind of an urban creek, a small creek that cut through that property. And here, this is a picture of one of the algal species that I found. This is a Platydarina. It's a colonial algal organism. And it's so cool the way it moves through the water. And I really, really enjoyed that project, and looking at all the different green algae and just different types of algae and getting to identify them. But it was really hard to identify algae. And so that was part of my duties at that internship. And then I also helped with some phenological monitoring of the restored prairies and woodlands there. And so that's where I really learned what phenology was. And I got more familiar with it, and I really liked tracking the progression throughout the summer of all the different things that would go through flowering and fruiting phases. And then shortly after that internship ended, I spent some time searching around for graduate schools that I wanted to do, and programs and advisors and types of research that I wanted to work on. And I ended up moving to Jonesboro in January 2020, and I started my master's at Arkansas State, and I started around the same time with Grace MacArthur. Some of you may have been to her talk last month, but we started at the same time so that we could work on lower Mississippi River islands together. And then last year, I adopted my cat, Jackie. It's her here. She's very cute. She's actually named Jackie after my favorite lower Mississippi River island, Jackson Point. So before I talk about my specific research, I also wanted to just talk about some of the research I did in the summer of 2020. I helped Grace MacArthur with her thesis. We did this flora together of 6 lower Mississippi River islands. I'm not going to go into the details and the results of that, because that's Grace's thesis research, and she did a whole talk over that was recorded, and you can watch that on the AMPS YouTube channel. But just wanted to show that I did go out, and it was a lot of fun getting to explore these 6 islands before I got to do my own research project in the summer of 2021. So throughout summer and fall 2020, I was out with Grace MacArthur and then Richard Abbott, and we would just go look for the plants on the islands, learn what was growing out there, and this kind of helped me develop my research project and to find species that I wanted to track the phenology of. And then in the spring of 2021, Brendan Kosnick joined us on the flora. You can see him here in that picture on the bottom. And then we just learned we were just discovering what spring plants were growing out there on the islands. So that was a lot of fun too, getting to just do a flora and learn about all kinds of plants and identify new species and everything that were new to the islands and new to the state and new to the county. That was a lot of fun. So just a little overview of my thesis research. I have 2 chapters. My first chapter, the overview, is to determine if inundation, which is correlated to elevation, and how the water levels reach the elevation of the islands, or of Buck Island, significantly influence phenology and plant fecundity, so that plant's ability to produce offspring. That's what I'm going to be talking about today, the results of that chapter. And then I have a second chapter, which focuses on scoring the phenology of herbarium specimens, so documenting biogeographic patterns of reproductive time through a latitudinal gradient, so a larger spatial scale, and then correlating those flower times with the different elevations of Buck Island. So I won't be talking about those, but just to give you a general idea of what my thesis research is. Okay, so getting into some background information about what plant phenology is. So phenology is the timing of events during the seasonal development of an organism. And for my project, I focused on plant reproductive phenology, which is sensitive to environmental factors like temperature, precipitation, and photoperiod. And because of that, it's often used as an indicator of climate change. So in temperate systems, a major focus of phenological shifts are changes in climatic conditions. So kind of these are some examples of common temperate system phenological studies with plant phenology. And so a lot of climate warming, soil warming because of climate change. I mean, photoperiod, but it's, I guess, not totally related to climate change, but just general things like that, environmental factors. But there are fewer studies covering the phenological shifts because of river flooding or inundation in these temperate systems. But that's not to say that there aren't studies that are being conducted on flooding and or riparian flooding and inundation on plant phenology, but I've noticed that a lot of them are mostly done in tropical forests where precipitation might be influencing phenology more so than flooding, like in a temperate system could be. Okay, and then some more background information about kind of the study system that I was in. So I worked on the Mississippi River, specifically the lower Mississippi River. And so the Mississippi River begins at Lake Itasca, and it starts as a small stream, so up in Minnesota. And then it cuts through the contiguous US and ends at the Gulf of Mexico. It's the largest river basin in North America, and then the 3rd largest river basin in the world behind the Amazon and Congo river basins. And it drains about 40% of the contiguous United States and portions of Canada. And I have divided, or the river's divided into 3 sections. And so I've kind of have drawn some boxes to show. So the upper river from Lake Itasca to the confluence with the Missouri River, that's the upper river. The middle, so from the Missouri River confluence down to the Ohio River confluence, and then the lower Mississippi River. So Ohio River confluence near Cairo, Illinois, to the Gulf of Mexico. And then the lower Mississippi River is a little different from the middle and upper, just because it's surrounded by the Mississippi alluvial plain. And it has historically meandered throughout the soft sediment of that river delta. And you can find evidence of that through old oxbows and river offshoots that are either still connected to the river or are cut off. But now this behavior of the river is being controlled through constructions of levees, dikes, dredging, and bank revetments that keep those banks from eroding. And then that also controls the depth and the flow of the channel for shipment transportation. And islands in the Mississippi River have developed from the river's natural meandering habit, but also many islands are formed as a result of steamboat wrecks and sedimentation following the construction of wing dikes. And then the lands surrounding the Mississippi River are pretty important to lots of wildlife. So about 60% of bird species use the river as a migratory path. And while we were out there, we've seen least terns, killdeer, kingfishers, blue herons, egrets, pelicans, seagulls. There's been a lot of bird sightings, so that's been pretty cool. Lots of wildlife as well. And then the Batcher, which is the lands between the levees, which includes the Mississippi or the river islands, that's mostly untouched, just because of most of the frequent flooding and the unpredictable nature of the islands or of the river. So these areas within the Batcher are relatively preserved and kind of like a safe space for plants and animals. So my study took place on Buck Island on the lower Mississippi River. And so the floodplain forests of the Delta have been relatively understudied, just because the land surrounding this area is used for row crop agriculture. And you can see on the picture on the left that all that light green color is just agricultural land, and then the dark green are forests. And then even lower Mississippi River islands have been understudied previously with respect to their flora, their communities, and their general ecology. And then here on the left, this is just kind of a snapshot of a portion of the lower Mississippi River where I worked. And here's Buck Island in red. And then I just have a better picture of it more up close. It's near Helena, West Helena, Arkansas, and it's found between the border of Arkansas and Mississippi. So Buck Island is a wildlife management area that's managed by the Arkansas Game and Fish Commission. It's about 3 miles long and 1,500 acres in area, and it's got some really cool habitats on it. So it's got some bottomland hardwood forests. You can see that on the top left. There's ephemeral swamps that have duckweed and water ferns. And you can see that here in the bottom left. And then there's willow forests, sandbars, mudflats, also a lot of vine thickets. So it's got some really cool habitats on this island, and can vary depending on where you are in the island. So the island has a pretty tragic origin. It started forming about 150 years ago after a deadly steamboat collision happened in 1865. So the Niagara sunk. There was a collision between two boats, and the Niagara, which was a steamboat, sunk, and 150 people that were on that ship drowned. And so it's thought that the sedimentation buildup after that steamboat wreck is what caused Buck Island to start forming. And it's also known as Prairie Point Towhead. So sometimes you might see that name pop up in historical maps. So right now it's public land, so it's used for hunting with permits and licenses, of course. And then there's also lots of recreational uses. Anyone can go to the island and hike around, hang out on the banks and swim. There's a company called the Quapaw Canoe Company that does guided tours to the island throughout the year as well. So it's a really beautiful island, and I definitely recommend checking out the Quapaw Canoe Company if you ever want to get a tour of a Mississippi River island and see some really, really cool and beautiful plants. So the Mississippi River experiences an annual flood cycle in which the river level is highest in the late winter and early spring, and then it's lowest in the late summer, early fall. And that's just because of the spring precipitation and snow melt from the north. So the islands on the lower Mississippi River have higher island elevations that will be not inundated or inundated for much shorter period compared to those lower elevations. And so plants at those higher elevations will be underwater for a shorter period of time than the lower elevations. And basically what I have here on this slide is just showing you a pattern of the river, so you don't have to pay attention to the exact numbers and things like that. But these are hydrographs that are provided by the Army Corps of Engineers, and it's just from 2017 up until fairly recent this year. And you can kind of see the general pattern where the river is higher in the winter, spring, and then starts to go down around June. And the spring flooding of the river is expected to increase with climate change, with warmer temperatures and more precipitation that will cause more frequent disturbance of the Bachelor and the surrounding floodplains as well. So there's going to be more flood disturbance in the future. So for my project, we had two main predictions. So the first prediction was that plants at lower elevations will have reproductive times that are later in the year compared to plants at higher elevations. And then plants at higher elevations will produce more fruits than plants at lower elevations. So they'll have higher fecundity. And that's because of a longer reproductive season from being less inundated and flooded on those higher elevations. So we established 62 habitat patches that ranged in elevation from 46 to 57 meters. And this picture here on the left, this is of on the right, this is of Buck Island, and it's a digital elevation model, and it just shows the locations of those habitat patches. And those patches were randomly selected from areas that had the highest elevation, so I could get the largest elevation range and kind of follow that gradient down. And then each habitat patch would be established in areas that were roughly in the same elevation and same habitat type. And each habitat patch would have 2 to 10 individuals in it. And then the picture on the left, that just shows the river bank, and then you can see how drastic that change in elevation can be right on those banks going up to the high elevations. And even here in this picture, those pink flags off in the distance, that's kind of what a habitat patch would look like. And that was probably a habitat patch of Malugo, which I will tell you about the species now. So from April through December 2021, we monitored 6 native herbaceous dicot taxa. So I had 3 annuals and 3 perennials. So I had Malugo verticillata, Roripa palustris, and Roripa sessiliflora. I combined those 2 and analyzed them at the genus level just because they had overlapping phenology, and they were hard to distinguish at the species level when they were sterile, and we wanted to tag these individuals before they started reproducing. And then I had Solanum emulans as the last annual. And then for the perennials, I had Oxalis stricta, Phyla lanceolata, and Viola sororia. So we would visit each individual and all the patches twice a week in the summer, and then once a week in the fall when the plants started slowing down on their growing. And every time we visited, we would count the number of buds, so the phenophases, so that would be things like the number of buds, open flowers, developing fruits, mature fruits. And we would do that for all individuals every time we visited Buck Island to do the phenological monitoring. And then pictured here on the left, top left, this is Matthew Jones and I, and we're counting those phenophases of a Malugo patch. And it would get pretty hot out there on the beach doing that. But usually the wind from the water would make us feel good, and we could hop in the river anytime we wanted if it got a little too hot too. And then on the bottom left, this is a picture of what a tagged individual would look like. So that's a Solanum plant, and you can see it has a unique identifier that will not overlap with any other plant. And then we would mark it with tape so we could see it again. So some lower elevation patches would go underwater during short periods of just higher river level, probably from just heavy rain events. And so that would pretty much scour those lower elevations of any plants, but sometimes the perennial species would be left. So like Phyla would be left, and you can see that here on this left-sided picture. Pretty much you can see where the water was and then receded, and what's left is some perennial species, but pretty much all the plants would be ripped away. But usually my tags would stick around. I only had a few tags, maybe a handful, that were lost to the river during that whole season. So then when that would happen, we would just wait for more plants to come up on those lower elevations, and then we tag them again and track them. And then this figure here, these are all the patches of Maluga verticillata. So the red dotted lines, those are the elevations of the habitat patches. And then the blue line is the river height throughout the 2021 season. And you can see that those habitat patches, at least the lower ones or the lower elevations, would be underwater as many as 4 to 5 times throughout the year. So because of these fluctuating river levels, I was pretty much tracking the river gauge at Helena every day and seeing how high the river was and determining which day would be best for us to go out. We mostly had a schedule of which days to go out, but sometimes we'd have to switch around based on how high the river was. So it would be pretty hard to get through that side channel. So not the main channel, but the one that was closest to the Arkansas side, that channel, when the river would go below 7 feet. So we know that we probably should wait until the river would go up a little bit more, or else we'd get stuck. So here on the right, this is a picture of Brendan and Travis pulling the boat through the water because we thought we could make it through a little inlet, and we got stuck. So many times we've had to get out in the middle of the river, and where it would just be just a little bit deep, we could still stand and push the boat through the water until we got through to a deeper section. And then on the right, that picture just shows that side channel. So that is a picture from the island looking at the mainland of Arkansas, and that those sandbars would be kind of hidden when the water would be just above them. And we've hit them a few times and sucked up sand and mud through our engine, and would have to take our boat in to get repaired. So I had to be pretty cautious about that and think about that before going out to the field. And also, if we would go out on days when the river would be really low, we know it would be a longer day because we'd probably have to get the paddles out at some point, and instead of being able to cruise up the channel. So there's a lot of mental preparation and boat preparation for going out to this island.
Hey, Caity, there's a question in the chat for
Oh,
you
yeah.
about the
Okay.
brown vertical marks on the charts, what they are.
So
From a
that's
few
probably
slides back.
from,
Yeah, I mean, we're typically we hold the questions till the end, but if you want to answer that one, go ahead.
Yeah, I know. I just was thinking, in order to have it make sense, if I think you've got some things coming up that are going to be similar.
okay. Which was it? This one probably.
That's what I was thinking.
Okay.
Maybe in that bottom center, there's that huge spike, like a misreading.
Yeah, I think that is a misreading. It's just a weird jump in the data. Like I'm sure the river did not. Well, I guess it could have. That's like a month. In February, it just went up really, really high, but it probably is a misreading. Yeah, these are just every day they would take an average river level and plot it. And so that's just one point is one day, one average for the day, or maybe it's I don't know if it's the average or maybe the highest marking of the day, but it's just one reading from the day. And sometimes there's weird things like that in their data. So I'm not really sure what that brown line is, other than maybe some error. Okay. So yeah, now I will talk a little bit about some of the results that I found. So for all of my statistical analyses and estimating any shifts in the phenology, I used the mature fruiting phenophase. So as predicted, plants at higher elevation had an earlier onset day of year than plants at lower elevations, except for Violaceuria. And so I'm looking at the figure on the left here. So that's the pink line, and that one just wasn't significantly its onset. So the day that it started to produce mature fruits wasn't influenced by elevation. And that's probably because it had such a small elevation range. It really was only found in wooded habitats and not on the beach like the other species were. But Oxalis had a different pattern. So that interaction was significant for that species. So higher elevations had a later onset. So when they would start producing mature fruits than the lower elevations, which is not what I predicted. And I think that might just be because those beachy lower elevation habitats, they got more sun than the wooded habitat. So there might be some other factor that's influencing the phenology of that species. Also, the elevation range is not as large for that species either, compared to the other 4 species. And then plants at higher elevations had an earlier peak day of year than plants at lower elevations, but just for Malugo and Solanum. So that's this figure on the right. And peak fruiting is the day in which the highest number of mature fruits were produced for each individual, and then average for the patch. And Malugo and Solanum both have the largest elevation range as well. And then I also did another linear model for cessation, so the ending of mature fruit development. And the only species that was significantly influenced, or that cessation was significantly influenced by elevation, was Solanum. So that's the only species that truly was shifted its onset, peak, and ending cessation. And there was not a pattern for all species while estimating the average number of reproductive days influenced by elevation. But so my prediction was that higher island elevations, the plants there would have more reproductive days or longer reproductive periods than the lower elevations, just because they had more time to be reproductive because those elevations were above water longer. But that was only true for Malugo and Phyla. So kind of mixed results for the species. And then my last model, I did a generalized linear model of the number of mature fruits and elevation. And so only Malugo, Phyla, and not Solanum, Malugo, Phyla, and Raripa, those species, so at higher elevations, their number of mature fruits increased. So there was a significant relationship between elevation and number of mature fruits produced. So 2 annuals and 1 perennial. So just to conclude on some of those results, the annuals were found in wider ranges of elevations than the perennials. So maybe there was just a higher signal between the relationship between elevation and the phenology for those species. But basically, my predictions were sort of supported, but it can't be true for all species. So I can't just say it's a pattern for all species. It's very species specific, basically. And then here's just another picture of that scouring that would happen, and my tag is knocked over. And that was also another interesting observation we noticed, because the water would look like it would be moving so slowly and calmly. But really it would be ripping up all of our plants. So that was kind of interesting. So just going to talk about now some of the interesting observations and fun stuff of doing this field work. So something that I learned was that oxalis has a pretty cool seed dispersal mechanism. If this video will play. Well, maybe it's not going to play. Well, sorry about that. It was a really cool video of the oxalis fruits whenever they're mature. If you just barely touch that capsule, those fruits will just pop out and go everywhere, and they'd like hit you in the face and everything too. So that was really cool. I had no idea that oxalis had that type of seed dispersal. And that's a bummer. We can't watch the video. Okay, but anyways, another interesting observation that we noticed, not just with my 6 study species or taxa, but with other plant species all over the island, is that the plants on those banks, those lower elevations, they would be much shorter than the plants like growing up in the woods and the higher elevations. And this is just a comparison. It's the second picture is not super helpful, I guess, because I don't have a ruler, but this is what a normal solanum plant would look like on the beach, or up on the higher elevations. It would be much taller, and next to the tag, it's almost the same height of the tag. And often those solanum plants would get much taller than the tag too, if you're familiar with this species at all. And then this is a mature solanum plant. You can see it has lots of fruits and flowers on it and is much shorter. And behind here is the tag, this rusty metal thing. So you can see they were much shorter, and they still would produce a lot of fruits and flowers too. So they were very reproductive. It's just much shorter. And we noticed that with many other species on the islands. Sometimes we would just be crawling around on the beaches looking at plants and think something was a different species, but it turns out it's just a weird, short plant of a species we were already familiar with. And then maybe that's just because those plants have sacrificed their height due to a shorter growing season. That might be what's happening there. Okay, now it's gonna play. Yeah, so that was just really cool to see happening. And then those I guess receptacles, the fruiting parts, would just be left on the plant, and so we just count them as mature fruits when we could tell. Usually it took us a moment to realize what we were looking at, the mature fruits compared to the developing fruits, and the mature fruits wouldn't have any seeds in there. But yeah, it was pretty cool. And so just some other adventures while doing this project. So we would be working in mosquito suits in the spring, so around April and May when this field work started. You can see this picture here. This is Grace and this is Matthew. They're probably collecting fruits, counting fruits off of a Raripa. I was collecting mature fruits at one point, or throughout the whole season, because at one point I thought I would need to count and weigh the mature fruits to kind of estimate the fitness or fecundity in that way. But so that's what they're doing here. And we would also sometimes have engine failure. We'd hit a sandbar, or sometimes our engine would just stop. And so in this top picture here, this is Matthew, Brendan, and Grace, and we're paddling back to the boat ramp because our engine is full of sand and will overheat if it can't pump water to cool the engine down. So we'd have some adventures like that, boat issues. We'd work through thunderstorms and rain. So here Grace, Brendan, and Matthew are taking a break while it's pouring down rain on us in the middle of counting a violet patch. And then we'd also be working through extreme heat in the sun and the heat of the summer. So we've got Brendan, Travis, and Matthew back there, and they're counting a Raripa patch. And then, of course, it's the Mississippi River. So we're dealing with giant flying Asian carp. So here's a video of that. Sorry if the audio was a little loud on that, but yeah, it was a lot of fun, and it's so beautiful out there on these islands. If you haven't ever been to an island or at least hung out in a bottomland floodplain forest of the Mississippi River, you really should go out there. And even near the boat ramp of Buck Island, there's a really cool river walk that you can do, and you can see Buck Island from there and go birding, and it's really amazing and beautiful. I definitely recommend everyone go out there and see it. So now I'd like to take some time and thank the people who helped me with this project. So I'd like to thank my committee members, Travis Marsico, Richard Abbott, and Scott Mangan. I'd also like to thank the Marsico Lab for their help in development of this project, but also all of the field work that went into it. So I'd like to thank Grace, Matthew, Brendan, Ben, and Diana. And then field assistant, so Tracy Klotz, he works at Arkansas State University. He helped us learn how to drive a boat and drive a trailer with a boat on it. And then also undergraduate student Maria Bollinger for her help on our last day on December 18th when we were out there. And then I'd also like to thank John Ruskey of the Quapaw Canoe Company for showing us how to access these islands, and then Johnny Waldrop with Buck Island Wildlife Management Area. He manages Buck Island. And then, of course, funding from Arkansas State University, the Superb Scholarship Program, the Arkansas Native Plant Society, and then the Arkansas Game and Fish Commission. And then I'd like to take some time to answer your questions, but I'd like to end with this slide. This bench has a pretty nice quote on it. This is from the Helena Tavern, which is just right by the river in downtown Helena. And it says, "This delta is a powerful land. It just leaps out at you. It seeps deep within you. The muddy Mississippi water leaves a stain on the soul that is virtually impossible to get, assuming any fool would try." So yeah, the delta is a beautiful place, and I loved working in it. I loved studying the plants there. As you can tell, I really loved it. This is Grace and I hugging Buck Island goodbye on December 18th. It was our last time out on a Mississippi River island, and we were going to miss doing this type of work. So it was really, really an amazing experience. Okay, now I'll take any of your questions.
Well, thank you, Caity. That was awesome. I've heard of tree huggers, but I think this is the first time I've heard of an island hugger. So that's amazing. So I like that last photo there. Yeah, if anyone has any questions, feel free to enter them into the chat, or you could probably just unmute your mic and ask. I know I had a question. I know some of the species you're looking at, Oxalis stricta, was it Gallium, Malugo, and then the other one I call it was Solanum, I'm guessing Carolinense or Carolinense. And then what were some of the others? So
it was Malugo verticillata.
Oh, Malugo. Okay.
Yeah. Oxalis stricta, Phylolanceolata, Viola soria, Solanum emulans, and then the two Raripa species. So I kept it not in the same family. They're distantly related.
Gotcha. Yeah, I got the carpet weed or the Malugo confused with, I guess there's a Gallium Malugo also, and I thought at first that that's what we were looking at. Okay. I didn't know if you happened to look at like the wetland indicator status of any of the species to kind of see, with the elevations, if any of them would have preferred like a wetter, closer to the or lower elevation habitat versus an upland.
Yeah, the flora, so the year before, I was kind of basing what species I'd pick based on what I remember to be common throughout the island, and I could find habitat patches of. And I think all of these species are wetland species, or species that could be found in wetlands because are weedy and can tolerate that. I would say the violet is probably the one that is kind of the odd one out. It's probably just more of a foresty kind of upland for a bottomland forest. And I think it's still probably, commonly found in wetlands. I mean, that species was common throughout our islands, so it can tolerate that habitat, but it was definitely the one that was a little different.
Gotcha. Yeah, I'm looking at for the Atlantic Gulf Coastal Plain region. I mean, it looks like some of them are rated as facultative upland and some as facultative.
Yeah.
So I just didn't know if you saw any relationship between that and how they might that might impact what elevations you might find them. So I mean, I think it's probably to where some of them are high up enough that it doesn't get inundated often enough, or the soil's not wet enough for them to be impacted, or they can thrive. But then some of them that are getting closer, like the solanum, it's probably gets wet enough to where it kind of stunts their growth, causing those to be shorter too.
Yeah, I think that would be an interesting thing to look into. I haven't done that or included that somehow in my analyses or in species selection.
That's fine. Yeah, I just didn't know if you found any relationship there.
No.
But, might be worth looking into. I can send you some info on that if that's anything you want to
Yes, please.
kind of look at. Okay.
Yeah.
I'll do that.
Whatever you're looking at right now.
Yeah, it's there's a spreadsheet you can get, and I'll send you the link for all where you can find it, as well as a spreadsheet itself, and you can kind of look up the indicator status for the species and see if you see any relationship there between where you're finding them, and might just be something else to. I mean, I hate to give you extra work at this point in your thesis, but it might be something to look at.
It's all useful. Yeah.
Yeah. Okay, cool. All right, let's see. Does anyone else have any questions? It's like Diane says, thanks for a very interesting presentation, up close and personal with the Mississippi River. It's not something we get to do in Northeast Texas, which I'm assuming that's where she's located.
Okay.
Lynn Jessup asks, real quick, Tim says, go Caity. And I guess that's from Jackie. And I'm assuming you know them.
Yes, that's Grace MacArthur's partner. They're watching at my apartment with my cat right now.
Gotcha.
They're cat sitting while I'm in Tennessee.
Oh, that's right. Your cat you named after the island. Yeah,
yes, Jackie,
which is awesome.
Yeah.
Yeah, I love that. That's awesome. Lynn asks, did you have to deal with any insects and or herps during your work?
Oh my, yes. Insects, yes. I got stung by yellow jackets for the first time last summer, like really bad. And I have apparently a bad reaction to them. The stings last for a few weeks and I get really bad hives. So we were dealing with yellow jackets. Sweat bees would be stinging us the whole time, sometimes when we would be counting the phenophases on the beach. So that was kind of irritating. Of course, mosquitoes, so many mosquitoes.
Can imagine.
In the woods. Yeah. And then sometimes ticks, but there really weren't a lot of ticks and chiggers on these islands. It's just mostly mosquitoes and then stinging insects.
I guess maybe ticks and chiggers might have difficulty making it out there. Is it just remote enough or isolated enough that?
Yeah, I mean, there's definitely wildlife. I forgot to mention that that there are a lot of wildlife on these islands. Like we see deer. It's pretty common to see deer and coyote. We'd see feral hog tracks all over the islands. Never actually saw one though. So I'm sure that they can be brought over.
Is it close enough to land to where when the Mississippi River level goes down enough, some of them might be able to make it over or swim over or get somehow inadvertently sent over there against their will?
Well, maybe. A lot of those islands are private hunting clubs or just private land used for hunting. But I think most of those mammals will either get over there through some sort of land bridge or wing dike or swim. I've heard feral hogs are pretty good swimmers.
Really? Okay. It's good to know if you're ever trying to get away from one, the water might not be the way to
I know.
like climbing like Travis here says, you saw a bear too. So I guess.
Oh my gosh, how did I forget about the bear? Yeah, thanks, Travis. That was the most exciting wildlife
bet.
sighting. Yeah. Yeah, Grace and I, we were alone on an island called Choctaw Island near Arkansas City. And we did see a bear at the base of a cottonwood tree. And that was
Wow.
why we were doing the flora. So
Huh.
that was really cool. It saw us and got scared and ran away. So nothing too scary there, but that was really cool. And then herps, we actually see a lot of box turtles and cottonmouth, lots of cottonmouth snakes. I think those are mainly it. We do have a lot of iNaturalist observations of these islands. Like there's, I don't know, if maybe some of you went to Grace's talk, but she talked a little bit about those, how we used iNaturalist for the flora. But we also were iNaturalisting insects and other animals and wildlife too. So if you just kind of went to Buck Island on the iNaturalist map, you'd probably see all kinds of cool things. The insect diversity on these islands
That's cool.
is amazing.
Yeah. Diane is asking about birds.
Yes, yeah. Saw lots of birds, least terns, killdeer, of course, blue herons, pelicans, seagulls, kingfishers, and then just lots of other bird sounds that we never really got any. Oh yeah, indigo buntings, Brennan says. Yeah. There was a lot of birds and a lot of cool bird sounds. We didn't really get some IDs on some of them, but it's amazing. You feel like you're separated from the rest of the world on these islands.
Yeah, like you're separated from the humans, but the nature is still there, you know.
Right.
I do wonder about like deer, you know, if for any islands where they might be a little isolated, if that would, the genetics, if they would, be inbred or anything like that, or if there's enough, genetic transfer between the island and other populations or not. But something to think about.
Yeah, maybe some other future mammal Mississippi River work. That would be cool.
Yeah. A little mammologist or grad student there. Travis says some of his favorite that he doesn't usually see were the filmy liverworts that grow on the mud in the shade, and lots of toads and or toadlets sometimes.
Yeah, right. I forgot about the toads. They were all over the place.
Diane asks, is there enough upland for the animals to be safe when the river is flooding?
I've wondered that before. We went out in April in 2021 and the water was pretty high. There was still land, probably enough for them to be safe. But I don't know. They might have to, I don't know. I'm not really sure what happens when the water would go up above the island. Those animals might not make it, or hopefully they have, I don't know, made it over to mainland before they got, I don't know, flooded away.
Yeah, or maybe they ended up on an island downstream. So
Yeah. It's a rough environment out there. Next to the river.
See, Travis says he's wondered if they can sense rising water and get out of there.
Yeah, they might be able to.
Well, any other questions before we wrap this up? Speak now or forever hold your peace.
Well, you can email me or contact me somehow too.
You want to put your email in the chat
Yes.
for folks? Okay.
Yeah.
Yep. Travis says, yay, great job. Such exciting work. And I agree. Thank you, Caity. This was awesome. I really enjoyed learning about your experiences and seeing all the fun stuff as well as what all went into it, and learning about just the journey to the island and during the low water, and then learning about your actual research itself. So really appreciate you sharing that with us. Appreciate you sharing your time as well. So thank you. Thank you.
Thank you for having me. This was a lot of fun. I like talking about the fun part of my research. I like talking about my research too, but it's fun to talk about the cool beauty of these islands and river.
Yep, I agree. I like it, especially the island hugging at the very end. Oh, well, thank you everyone again for being here with us today. Again, this recording will be placed on our YouTube channel, so please check that out. I'll try to get that up there sooner than later, probably here shortly if I'm able to today. Our next webinar coming up is going to be Saturday, September 3rd at 10 a.m. We're going to learn about the biodiversity and role of biodiversity and the role of disturbance in managing natural ecosystems. That's going to be with Nate Weston of the Beaver Watershed Alliance. He's currently also the president of the Arkansas Native Plant Society, so that should be a good one. Tune in for that. You can find out more information about the Arkansas Native Plant Society on our website, anps.org, or you can follow us on Facebook at facebook.com/Arkansas Native Plant Society. I encourage you to go subscribe to our YouTube channel. Just type in Arkansas Native Plant Society on YouTube, and you can find all of our past webinars as well as this one here. Again, thank you, Caity, for being with us today. We really appreciate you and learning about all the great work that you're doing. And again, to the folks out there, if you're looking to hire a bright young botanist, and I'm sure Caity would appreciate getting a call from you sometime before December. So thank you all. Have a good day, rest of your weekend.